Researchers from Museums Victoria are cryo-freezing cells from 100 species in the hope of conserving some of Australia's most vulnerable species. The Living Biobank involves storing cells at temperatures around minus 196 degrees Celsius. Joining us live with more is Dr. Joanna Sumner from Genetic Resources at Museums Victoria. Joanna, thank you for your time. Appreciate it. Tell us a bit more about why you're doing this and how this could actually help your cause of conservation one day.
Well, our aim at the moment is to start to conserve live cells from wildlife. So this is something we've been doing for years with human research. So it's similar to IVF in that we're freezing down sperm, eggs and living cells from wildlife. So using the same technology that's been used for humans and in livestock as well. But I guess our aim is to halt or slow down the biodiversity loss that we've seen worldwide.
Where is the technology up to in terms of trying to recreate a species that may go extinct, for example?
So there's a fair bit of work has been done with small mammals. So Australia actually has one of the worst extinction rates for small mammals in the world. So a lot of work has already been started. So the University of Melbourne is one of our partners in this project, and they've been working with some small mammals and marsupials and working to grow living cells in a way that they can be put back into the wild, so that we can start to really make a difference to some conservation programs. This is an additional way we can start to conserve our unique, amazing wildlife that we have in Australia. And I guess we've started with marsupials, but we're also doing some work with reptiles. In Victoria, there was a recent rediscovery of the Victorian grassland earless dragon that hadn't been seen in 50 years. But we were able to take a tiny bit of the tail of one of these newly discovered individuals without doing any damage to the animal itself and grow some cells. In fact, grow enough that we could store some into our cryo tank, and that will be there for hundreds of years or in perpetuity. We also were able to sequence the entire genome of that animal from just the tiny cells on the tip of its tail.
Goodness, we were just watching some images of what looked like a pot of freezing material. Just tell us a bit more about what that process actually is, how that works, what you have to do to actually make it happen.
So that is footage from our biobank at the Museums Victoria Research Institute, and we can see our postdoctoral researcher, Dr. Jennifer Hetz there. She is currently putting some cells that have been grown in a special medium, and those are now going to be put into the biobank. So the big tank it's going into has liquid nitrogen at the bottom, and you can see all of that vapor in there that keeps those cells at minus 196 degrees, and that pretty much stops all biological activity. So the cells won't change at all, but when we take them out, whenever we need them for ongoing research, we thaw them in a way that the cells can then grow and divide again. And those cells will be available for research at the Museums Victoria Research Institute or University of Melbourne. And we also have collaborators at the Australian Museum in New South Wales as well, and we're all working together to be able to use these little bits of DNA or little cells that they're growing to get that genetic diversity back into wild populations, particularly with threatened species.
Dr. Joanna Sumner, it all sounds very sci-fi, but really amazing stuff. Really appreciate you talking us through it. Thank you.
Thank you. We really hope we will make a difference to conserving our extraordinary biodiversity with this program. So thank you very much for having me on.